Okay, let's go through the ear and the eye development really fast, or as fast as I'm able to. So on day 22, the anterior neuropore hasn't quite finished closing. It's starting to close. Remember, it closes on day 25. It starts having like outgrowths or pouches, and those are going to be your future optic vesicles. The neuroectoderm is going to eventually make the retina, the posterior layer of the iris, and the optic nerve. The lens placodes, on the other hand, are going to be made out of the surface ectoderm, and those will eventually give rise to the lens and the corneal epithelium. So as you can see here, it invaginates in and then eventually forms the hollow circle or sphere. Sorry. The groove will form, which is the optic cup, and then the optic stalk, and the lens placodes will invaginate, making a two-layered pit. The mesenchyme will migrate into the retinal fissure, and that makes the blood supply along with the fibrous part of the eye. And then the neurocrest cells will migrate in and make the choroid, the sclera, and the cornea. So here is your ectoderm. Here is your optic vesicle. It invaginates in, forms a cup, and this closes. The cornea epithelia will come in from the neurocrest cells, and then eventually it will form the lens capsule and the layered corneal epithelium. The optic groove will come in and form in the neurofolds from the future diencephalon. The axons will enter the optic stack and make the optic nerve, and then the lens pit will form when the lens placode invaginates into the surface ectoderm, and then the center of the optic cup will form the optic disc. Optic cup will form the retina, and the optic stalk will become the optic nerve. Mesenchyme will fill the choroid fissure to make the hyaloid artery, and that will eventually make the central artery and vein of the retina. There's only one. As you can see here, lens comes in, optic stalk starting to form the optic nerve, and then the central vein and artery will divide into the proximal and distal part. The distal part will degenerate and make the hyaloid canal, while the proximal will remain. That's why there's only one artery and one vein draining and supplying the retina. The outermost layer is going to be pigmented by melanin. That usually appears by the 6th week of development. And the inner is going to be forming the retinal neuroepithelium. And the layers are separated by the intraretinal space, which closes but doesn't fuse. And the neuroepithelium will form the rods and the cones, the bipolar cells, and the ganglion cells. And then the neuroretina is going to be transparent. The lens placode, once again, invaginates, and it separates from the surface ectoderm and becomes a hollow lens vesicle surrounded by a basal lamina. And the mesenchyme cells in the lentiretinal space will make the primary vitreous body. By week 7, the posterior cuboidal cells elongate into the columnar cells that will obliterate the lens vesicle, making the primary lens body. And the anterior wall cells will proliferate throughout life because the eye always needs to replace the corneal epithelium. By the 3rd month, the cells will migrate peripherally to the lens equator, and that will make the secondary fetal and the adult cortical lens fibers, also known as the lens bow. The choroid and the sclera will form in the weeks 6 to 7. The mesenchyme around the optic cup will differentiate into 2 layers: the inner pigment layer, which is the choroid, and the outer fibrous layer, which is the sclera. The leptomeninges, remember in the spinal cord was the pia and the arachnoid. In the eye, it's the choroid and the dura, which will make the sclera. The choroid is made out of the neurocrest cells, which are the stromal cells, melanocytes, pericytes, and the mesoderm is going to be made from the endothelial, or it's going to make the endothelial cells of the vessel. The anterior sclera begins as a condensation of mesenchymal tissue that is going to be continuous with the cornea. And by the 12th week, the mesenchymal condensation will reach the optic nerve. Cornea is going to form once it's induced by the lens vesicle, and the external corneal surface is going to be made out of the surface ectoderm. And the mesenchyme is going to be forming the substantia propria and the endothelium. The stroma is formed from the stratified squamous epithelium, the endothelium of the vessel, and the Bowman's layer. Now here's the surface epithelium, mesenchymal tissue coming in, and the neurocrest cells will make the keratinocytes and the stromal cells of the eye. So here's the pigmented layer, the vitreous is forming. Eventually, it will kind of solidify in this kind of gelatinous kind of form. Retina is forming right here. Choroid supplying, and the sclera. The iris will form from the rim of the optic cup, and the neurocrest cells will make the connective tissue stroma. The neuroectoderm will make the dilator and the sphincter pupillae, and the ciliary body will develop from the optic cup, but it's posterior to the iris. The lens is suspended from the ciliary bodies by the suspensory ligament, and the mesenchymal cells will make the ciliary muscles. Ciliary muscles are important in accommodation, so focusing of the eye. Week 6 is when the fold of the ectoderm come close together and they fuse in week 8. The space between the lid and the cornea is also known as the conjunctival sac, and the lacrimal glands will form from invagination of the ectoderm at the superolateral angles of the conjunctival sac, and they mature about 6 weeks after birth. The lid muscle, of course, comes from the mesoderm tissue, and the nasal lacrimal furrow will make the nasal lacrimal duct. So there's a bunch of defects of the eyes. Coloboma is going to be a problem with fusion. It's usually a mutation of the PAX2 gene, and it is involved in incomplete closure of the retinal fissure. So there can be a retinochoroidal coloboma, a palpable coloboma, which is where the lids don't fuse correctly, coloboma of the iris, where there's going to be like a keyhole kind of shape of the eye, and cyclopia, there's going to be a loss of midline tissue, and there is an unnatural fusion. Other conditions would be microphthalmia with small eyes, anophthalmia, no eyes, aniridia, no iris, and then there's conjunctival cataracts. Okay, so during week 4, the ear forms, and it comes, the optic placode is formed from the neuroectoderm. The optic placode will sink into the mesenchyme, gets invaginated, makes the otic for ear, optic for eyes. Optic placode forms the pit, and then eventually it forms the vesicle. The vestibular part of the ear, or the endolymphatic sac, will form from the otic vesicle, and that will make the utricle, the saccule, and the semicircular canals. The cochlear part will make the ducts and the coils. The 3 diverticula will grow from the utricle parts of the membranous labyrinth, and the central parts of these diverticula fuse and disappear, and the peripheral part will become the semicircular ducts. Ampulla will form from the ends of the semicircular duct, and the cristae ampullaris will differentiate in the ampullar and the utricle and the saccule. The saccule part will form a tubular diverticulum, and that makes the membranous cochlea. In the inner ear that forms in the weeks 20 to 22, the organ of Corti will differentiate from the cells on the wall of the cochlear duct. The ganglion cells of cranial nerve 8 will migrate in to make the bipolar neurons, and that makes the spiral ganglion. And it extends from the ganglion to the hair cells. The bony labyrinth is the mesenchyme that differentiates into the cartilaginous otic capsule. The vacuoles will appear inside and coalesce to make the perilymphatic space, which will eventually form the scala tympani and the scala vestibuli. And the cartilaginous otic capsule will ossify and the bones will form. And the PAX2 gene is going to be responsible for making the spiral organ of corti and the spiral ganglion. So inner ear, weeks 20 to 22, this is all responsible for our sense of balance. Okay, so middle ear is going to be formed from the endoderm. That makes the tubotympanic recess of the first pharyngeal pouch, and the ectoderm, which is going to form the outer ear. Those grow towards each other, so you can see here. And the pharyngotympanic tube is going to connect the pharynx to the tympanic area, and it's known as the eustachian tube as we grow up. And the first pharyngeal arch will form the malleus and the incus, and the second pharyngeal arch, along with the neurocrest cells, will form the stapes. The tensor tympani is the muscle of the inner ear or of the middle ear and allows you to like pop the ear to equilibrate the pressure. It's formed from the mesenchyme from the first pharyngeal arch, and in some people it's supplied by cranial nerve 5. And why is that? Is because some people have it under voluntary control, some people don't. That's why people who do not have it under voluntary control can't go snorkeling, or not snorkeling, scuba diving, because they're unable to equalize the pressure and can rupture their ear. The stapedius is going to form from the second pharyngeal arch and is supplied by cranial nerve 7. External ear comes from the mesoderm, and it develops from the dorsal part of the first pharyngeal groove. The ectodermal cells at the bottom of the primordial acoustic meatus will make a solid meatal plug, and then it degenerates in late fetal life. And children can hear a lot better than adults because the meatus is short until it reaches the adult length at 9 years. Children can hear like higher pitched noises a lot better too. That's why, because the meatus is short. So if you whisper anything around children, chances are they're going to pick it up. The tympanic membrane's outer layer, which is the ectoderm, and it forms from the first pharyngeal groove. The tympanic inner layer is going to form from the endoderm of the first pharyngeal groove pouch. And the mesenchyme of the first and second pharyngeal arches will play a role in formation, while the auricular helix from the first and third pharyngeal arch will surround the first pharyngeal groove. And the auricle will develop from the base of the neck and it will rise. So a lot of genetic disorders have one of the signs is that the child will be born with very low hanging ears. Like the ears is like at the neck, really. Okay, so hearing loss is correlated to a mutation in the GJB2 gene, and that can be caused by rubella or cytomegalovirus infection. Isotretinoin use, so vitamin A. So if anybody uses like tretinoin or takes medication for acne, it can develop the first arch syndrome, and the patients will have microtia or anotia. Sorry, not really sure how to pronounce it, but that means tiny ears or no ears at all. And there could be an accessory auricular heliform or peri-auricular sinuses and fistula. So the fistula would be like that kind of dot right there. And of course, it depends on the penetrance. Grade 1 is not too bad. Grade 4 is the worst. It's a complete absence of the ear and the canal. That's it for now. Thank you for your attention, and good luck studying.